gives me great pleasure to introduce our initial speaker, because as you're going to see, we're going to begin now with a brilliant scientist that will present his research of quite a number of years on this particular topic. I've had the great pleasure of lecturing with Dr. Ritchie, and I can tell you that you're in for a treat as a kickoff speaker for this morning, because he will lay the foundation for what is to follow. Dr. Ritchie is an associate professor in the Department of Biomaterials and Biomimetics at NYU College of Dentistry. He graduated from Muhlenberg College in Pennsylvania in 1977 with a Bachelor of Science degree, and subsequently earned his PhD from UMDNJ in '84. He has a number of research interests, one of which is the development of controlled microtexture surfaces for dental implants that promote bone, connective tissue, and epithelial attachment. He is one of the developers of the laser-lock microtexture surface. He is also working on the development of three-dimensionally printed scaffolds consisting of permanent and resorbable materials, another exciting topic down the road, another great technology for replacement and regeneration of craniofacial bone and soft tissue structures. He is well published, and I can tell you that this is a gentleman that brings very esoteric scientific material and distills it in such a manner that we can all understand it. It gives me great pleasure to introduce Dr. Jack Ritchie.
Thank you very much, Mike. I'm very excited and very proud to be here today. Also a little bit humbled because as a basic scientist, it's not often that you get a chance to do this. I was trained really as a cell biologist, anatomist, and materials scientist. And most of the time, when you're a researcher like I am, you don't get a chance to see your projects actually turn into something real that's a real product. And this is something that I've been working on literally for 20 years. So this is really a lot of fun for me today. The only thing that's a little scary about it is the people I'm up here with Mike Picos and Myron Evans and Stu Fromm. It's like, it's a little bit scary company to be here with. Anyway, what I'm going to start with today is literally the science behind the laser lock microchannel surface. This is a project that started an awfully long time ago, and it started the right way, and I'm going to show you a little bit how things start the wrong way. This comes to me by John Brunsky, who lectures for a master's program that we do in biomaterial science, and I thought it was appropriate. The way we're supposed to develop biomedical devices and the way we teach our students that it's supposed to be done is you're supposed to start with a clinical problem, and you're supposed to do some engineering and prototype development, and there's a logical progression that's supposed to happen in this process. And you're supposed to do animal studies and refine the product, and then go through a clinical trial, and then marketing, and then application eventually. And unfortunately, with dental implant surfaces, we haven't always followed that plan, and most of the things that are on the market, and I'm being a little cynical about this, are essentially marketing-driven surfaces. We find something that's a cool-looking surface. Then we find a way to mass produce it, put it on an implant. We make lots of them and get FDA approval. And getting FDA approval for an implant is not that hard as long as you're not trying to get any claims from the FDA, at least here. You sell a lot of them, and then eventually you do some animal studies so you can back into some science to support your marketing, and you end with a clinical trial that shows at least a 95% success rate. And this is a rule that is present pretty much in dentistry as well as orthopedics. I spent 16 years in the orthopedics field, and there's some rules that cross the lines between both fields. And one of them is that, in general, you don't publish too many clinical trials that show less than a 95% success rate. You occasionally see a 93 or something like that. But unfortunately, that's the way things have worked in this field, which is really not very science-driven. And what it's led to is some rather interesting designs and some things that we've learned a lot from. I used to run the implant retrieval lab at Hospital for Joint Diseases when I was in orthopedics. So we used to see all the interesting things that fell out of people for one reason or another, and you can learn an awful lot from your mistakes. And we saw a lot of mistakes. And actually, a few of them are here. And some of this stuff just, there wasn't very good science behind some of it. This is actually from an '88 cover from the Journal of the California Dental Association. Even in 1988, there were so many different implants around with, different thread designs and different concepts, cylinders, HA-coated cylinders, single crystal sapphire implants, all kinds of wild stuff that was around. And very little of it has survived the test of time. Certainly some of the implants were interesting concepts. The Zircon implant, what we call the Mollybolt implant, the old blade implants were an interesting design, and they were the beginning of the current implants, but they've been replaced too because they're a little too surgically intensive and they didn't last very long sometimes. This one's one of my favorites because this one kind of comes out of the orthopedics field. In orthopedics, when you do intramedullary rods, they very often use a fluoroscope to put pins in the ends of them to fix them. And this is a dental implant that had a fixture that fit on the collar that allowed you, it had a drill guide, so you could put pins in to hold it in place. The only problem is it was a little bit cumbersome trying to fit all that in the patient's mouth, and it was a little bit surgically invasive, to say the least. The way we developed laser lock was different, and it was because it started as a basic science project. What happened is we started with a clinical question in 1988. Basically, we said, how does texturing work? We knew that texturing produced a different response than non-textured surfaces. And so we started doing some very basic science studies to look at how cells respond to textured surfaces. We then started in the early 90s trying to transfer that technology, what we found, the surfaces we identified, onto metal and make metal prototypes. And we then began doing what are called animal screening studies. And when I say screening studies, I mean studies that are designed to test multiple surfaces and see which ones work the best. We then did the refinement part of that where we put the surface on a dental implant, and we did what's called an animal application study where we tested them in animals. Then we started human clinical trials, and this was originally done with BioLock, which we started working with in 2000. The implant received FDA approval after clinical trials and the animal data in 2004, and then BioLock was acquired by BioHorizons. And BioHorizons has done more refinement on the surfaces, has produced some very interesting implant designs, and really what has come out of this is what I would call the next generation laser lock implants, the next generation surfaces and the implants. And as you'll see, these are very interesting, and they're going to be very successful. What we started with is this implant on the left. This is the BioLock Silhouette implant. It has, you can see it's a tapered implant. It has an interesting thread design called a reverse buttress thread that puts compressive load on the bone. It has this tissue engineered collar that's mainly what I'm going to talk about today. That's the laser lock collar. And it has what BioLock call an Osteolock surface, which is a high roughness, clean blasted surface. Now, what that has led to, of course, is the laser lock tapered internal implant by BioHorizons, which you'll notice is very similar. The thread design is similar. The main difference is the collar portion of the implant now has BioHorizons internal connection. And other than that, the surfaces are the same. One of the interesting things about the acquisition of BioLock by BioHorizons was that the companies had very similar philosophies. It was a tremendous fit for BioLock to be part of BioHorizons because we even use the same surface. This surface is actually produced using the same blast medium for the two implants. I mean, there's a lot of things that we shared philosophically, and so it was a very good fit. And this is a very nice implant, the tapered internal. Now, we've seen a lot of surfaces come and go. We've started, of course, very early on with the original machine surface implants, the original Branamark surfaces. We know from a long time and from very empirical data that we know that smooth surfaces will not integrate in bone. We know this in orthopedics, we know this in dentistry. If you know anything about fracture fixation devices, you know that fracture fixation devices like plates and screws and intramedullary rods are all polished. And the reason for that is you want to be able to take them out later. If you were to texture them, you'd never get them out of the patient. So, empirically, we've known about texturing for a long time, but we just haven't really taken advantage of making texturing work the way it can work. And so we've gone through a series of surfaces. The original blast textured surfaces were blast textured with corundum and some materials that were not so good and stuck in the surfaces, and that was what led to acid etched surfaces originally, because you had to acid etch them to clean them. We then developed combination surfaces such as the sandblasted acid etched surface, the resorbable blast textured surface, and the anodic oxidation surface. But the point I want to make is these are all random textured surfaces. You can control to some degree the roughness, and you can control the range of structure size, but you can't control the organization of the surface. The laser lock surface is something different. This is the laser lock surface. It's the first micro-engineered surface that is organized and produced to control the way cells interact with the surface. These grooves are, in this case, 12 microns wide, and the ridges are 12 microns wide. And so this is actually, if you look at comparable size scales here, you can see that we're dealing with a size scale that's comparable to the texturing on other implants, but it's an organized microtexture. It's produced using a laser machining method, and every one of these surfaces looks exactly like the rest of them. They're very reproducible. Now, how did we arrive at that surface? Well, we started out, as I mentioned, a long time ago in 1988, looking at how texturing works. We were looking at textured surfaces that osseointegrate versus smooth surfaces that were adjacent. We had a smooth metal surface, and all you saw is a fibrous capsule that would form next to it, even if they were in the same animal in the same surgical site. And if there's a general rule that crosses in orthopedics and dentistry is that textured surfaces osseointegrate while smooth surfaces fibrointegrate. And that's something that we've published some papers on, and we're not the only ones, certainly. And in looking at the cell response to it, there are a number of labs that have looked at cell response to textured surfaces. Barbara Boyan's group and Zvi Schwartz have done a lot of work on this and looked at what different structured surfaces do to cell, basically cell response to circulating growth factors and vitamin D and different bioactive agents. They've come to the conclusion that unfortunately most surfaces are mixed topography surfaces where the cells will find an area that they like, but some cells perhaps don't. And that there are certain values that we should be looking for. An RA, a roughness height value of greater than 2, an RM, a spacing value that's around 10 microns. And actually their results agree somewhat with what we've found. This is a new paper, actually, that just came out actually in 2007 that also shows something that's very important with textured surfaces. The two slides on the pictures on the right are cells grown on a smooth Thermonox surface, which is a culture plate. The two surfaces on the two slides on the left show cells growing on an SLA surface, a sandblasted acid etched surface. And essentially what they found, and this is what we've seen too, is that textured surfaces inhibit the spreading of cells. The cells will not spread on the textured surfaces as well as they do on a smooth surface, and it turns out that's very important. Now, we did some studies back a few years where we actually textured culture plates. This is a culture plate that part of it was masked. This side is smooth. This side was blast textured. And we used this model. This is called a dot colony model, where we put about 20,000 cells in a microscopic collagen droplet, about 2 microliters, and we polymerize that on the surface. These cells then grow out of that microdroplet and form a colony. And by measuring the rate and size of the colony formation, you can see how the cells respond to different types of surfaces. And you can see here, now this surface, even though one side was blasted, we vapor deposited titanium oxide on the entire surface. So all the surface chemistry is the same on that surface, but the topography is different. These colonies were all started with the same number of cells, but you can see the colonies on the smooth surface grew a lot faster than the colonies on the textured surface. And even when we look at cell density, the cell densities are even less on this side than they are here. So it's not like the cells are just bunched up on the textured side. They simply do not grow as fast. And so we started to look at how to make controlled textures. And at that time, about the only way to make a controlled texture was to use silicon wafer technology like they do in microelectronics. So you see this silicon wafer has a bunch of blocks on it. Those are actually different patterns that are made using photoresist and reactive ion etching. And you can create a template that you can then cast culture plates from. And so we made our own culture plates that had different size and shape microstructures on them. We vapor deposited them with titanium to simulate an implant surface. And then we started looking at how cells responded to these surfaces. This slide shows on an individual level how cells respond to a microgroove surface like this. This is actually one of the cast culture plate surfaces. When you grew cells on them, the individual cells would sit either within the grooves or on tops of the grooves. And when you looked at the overall colony formation, instead of forming circular colonies, these formed elongated colonies. And while you can't see the microgrooves here because they're too small, the grooves are running in this direction. And the colonies grew along the microgrooves. But they only did this if you worked in a very specific size range. You had to work in a size range between 6 and 12 microns in terms of the structure size to really get this to work well. If you were below about 4 microns, the cells start to bridge the grooves and you don't get as effective a response. And if you get above about 20 microns, the facets of the surface are big enough for cells to spread enough that they start to act like they do on a smooth surface. So we identified this size range of structures that work very well. And what we found, and actually we just published kind of a compendium of a whole bunch of experiments that we did on this as a part 1, part 2 paper that's in the Journal of Biomedical Materials Research with a colleague of mine, John Grew, and one of my mentors, Harold Alexander. And part of this is on how colonies grow on these microgroove substrates. Part of it is on how the colonies grow, how cells deal with this on an individual level. And what we found, of course, is that the cells orient along these microchannels, and that is based on the way cells send out pseudopodia and the way cells actually explore substrates. What we found, just to summarize these papers, is that the colonies organize along the microchannels on a colony level. They grow parallel to the microchannels, and you can actually inhibit fibrous tissue growth by at least 50% just using the microstructure of these surfaces compared to flat surfaces. On an individual cell level, the microchannels control cell shape and control cell spreading. And we'll talk about the importance of that in a minute. And that's what this graph is. This is cell area of attachment on a smooth surface and then on some of the microgroove surfaces. And you can see the area of attachment is much lower for cells growing on the microgrooves. And again, most of the best response we saw in general with the control of these cells is in this 6 to 12 micron range. Now, why is spreading important? Well, part of this is based on what's called contact guidance. Why cells respond to microstructure the way they do has to do with the way cells explore substrates. Without getting into the science of it too heavily, because there wouldn't be time, and this isn't the right audience to go into cell tensegrity theory, but there are reasons why cells send out pseudopodia basically in straight lines when they explore substrates. And if the pseudopodia that they send out runs into a structure, it stops, and they send out a pseudopodia in another direction. So what happens is, and this was noted a long time ago, one of the most famous biologists around the turn of the century was a fellow named R.G. Harrison, who was the first person who ever grew nerve cells in culture. And he was the first person to actually publish papers suggesting that cells would migrate along microstructure, because in the days when in vitro meant in glass, they would reuse culture plates. And if it was cheap glass and you scored the culture plate up with an abrasive, they noticed that cells would hit a microstructure like a groove, and the cells would migrate along the groove. And R.G. Harrison noted this in 1907. Paul Weiss, who was a very famous cell biologist, published a paper on this in 1952, and he's the one who coined the term contact guidance. Contact guidance turns out to be a very important concept in how cells respond to microstructure. It's probably the reason cells respond to texturing the way they have in the past. We've just never really optimized it before. This paper, by the way, was just republished by the National Academy of Sciences because it's actually one of the first papers on tissue engineering, although it wasn't called that then. Now, one of the things we noted, too, that involves the contact guidance was that epithelial cells also respond to these microchannels. And this is what got us directly involved in dental implants. If you look at this colony of bronchial epithelial cells, we started the colony here and they grew out onto a flat surface. This colony was started on a smooth surface that's next to an 8-micron microgroove surface. And you see that when the cells reach the 8-micron microgrooves, they bunch up on the edge of those grooves and they start growing along the grooves. And they have a lot of trouble crossing those grooves. And this concept was what taught us that we could use this surface as a barrier for epithelial migration. And this is what got us directly involved with dental implants. Because up until then, we had really been looking at the way we could possibly make surfaces for orthopedic implants. Until then, I was in the orthopedics field. So this is contact guidance again on a colony level. These cells are reacting like these cells. They're running into a structure, and they're migrating along it rather than across it. Now, why does that change anything other than migration? Well, if you take this cell, that's normally the way cells grow on a flat surface is they spread out a lot, and they form a lot of cell attachment plaques, otherwise known as hemidesmosomes. These are actually fluorescent images of the hemidesmosomal formation in one cell. If you grow them in a microchannel, they look like this. And what that does is it limits the cell spreading. By the way, there's another way you can do it. You can do it with a lot of small microstructure that limits where the cell can attach. The problem with that is, and that's probably one of the things we've been doing with microstructured surfaces, is that this is not directional. You can't control the orientation of the cells that way. This way you can, by putting them in a microchannel. And what that does, I mentioned earlier that the microchannels limited cell spreading. Well, back in 1978, Judah Folkman, who was a pioneer cancer cell biologist, was looking for ways to control cell growth. And he found that by limiting the way cells spread on surfaces, and he used attachment proteins to do this, either a lack of or by adding attachment proteins, you could control the way cells attach to surfaces. And if they didn't attach, especially mesenchymal cells, fibroblasts, bone cells, those types of cells, if they don't attach, they undergo cell death. And when he did this, that wasn't called apoptosis, which is programmed cell death, but they knew that the cells, if they didn't attach, they died. Even if you got some very minimal attachment, you could get cell death. But if you could get a reasonable amount of attachment but limit the spreading, you could keep the cells in a differentiated state, meaning they would perform, they would make extracellular matrix, but they wouldn't proliferate. And a fully spread cell, on the other hand, goes into proliferative mode. I won't go into the mechanisms behind this, but it has to do with the amount of cell attachment plaque formation, which is a form of ligand binding, which stimulates cell proliferation. So by limiting cell spreading, which is what we're doing with the microchannels, we can limit proliferation of cells, and in particular, we can prevent fibrous encapsulation of the surface.